Okay, good afternoon and welcome. We're glad you can join us for this first session of Explore Caltech 10-minute science talks. I'm John Bostic, a postdoc at Caltech, and I'm joined here today by my postdoc colleague, Pablo Garrido Barros. We are both co-chairs of Outreach in the Caltech Postdoc Association, and we thank you for joining us today. Explore Caltech is an outreach event between Caltech and our community. Although the in-person Explore Caltech event has been canceled this year, we wanted to allow the Caltech community to have an outreach event with the community online. We thank everyone who has made this possible. A special thank you to the speakers today and the speakers who will be presenting on Wednesdays at 5 p.m. throughout the month of May. Before we begin, I would like to mention that after each of the 10-minute talks, we will have a brief question and answer session using Zoom chat. If you have a question, use the Zoom chat function and send me a message, the host, John Bostic, and we will ask those questions after the end of the talks. So, and one additional thing, during the presentations, please keep your video off and your microphones muted so as not to distract the presenters. And now, Pablo will introduce the first speaker.
Hello, everyone. Well, our first speaker today is going to be Kaichuang Yang, who is a former graduate student in the chemical engineering department, working in the design and engineering of one of the most important molecules for life, that is proteins. He was a member in the group of Professor Frances Arnold, who was in fact awarded with the Nobel Prize in Chemistry in 2018. Currently, Kevin is a senior researcher at Microsoft Research in New England, where he is working in the computational designs of new proteins. Kevin, thank you very much for joining us today and looking forward to your talk.
Thanks, Pablo. I'm going to share my screen. Okay. Well, thanks, Pablo, for the introduction. So I'm going to talk about protein engineering, just the art and science of redesigning the molecules biology uses to do everything. So as a human, each of our cells contains between 1 and 3 billion proteins each. These include, for example, collagen, which is a protein that makes up the structure in your skin. They include antibodies, which are proteins in your immune system that help find and fight off infections. Hemoglobin is a protein in your red blood cells that carries oxygen. Insulin is a very small protein that tells our body whether to keep more sugar in the blood or to put it into cells where it can be used for energy. A more recent example of a very relevant protein is the SARS-CoV-2 spike glycoprotein, which is the protein in SARS-CoV-2 that allows it to enter your lung cells and cause infections. And another fun example is luciferase, which is the protein that makes firefly butts glow. So given that wait, can you all see my screen?
Hey, Kevin, we can see your screen, but I think it's stuck in the first slide.
Okay.
And also there is a window saying "will order."
Okay, give me a second here. Okay, does the new slide show up now?
Mm. Now it's a different slide, but
Okay.
yeah, I think it's working now.
All right, so given all the proteins I showed you before that already exist in nature, like collagen, like antibodies, hemoglobin, insulin, that spike glycoprotein that allows the SARS-CoV-2 virus to infect your cells, and luciferase, which makes firefly buds glow, why might we want to design new proteins? Well, for example, we might want to do some new chemistry. We make and use lots and lots of plastic bottles, plastic bags, and those bottles, if we throw them away, don't break down. They last pretty much forever. But chemically, we can break those down and recycle them. But what if we want to do that with a protein? Well, then you have to design a protein that will do that, and people are working on that. A lot of important new drugs are proteins. For example, this is asparaginase, which is a drug used in chemotherapy. Sometimes scientists try to redesign proteins to learn how they work. You might change the protein and see whether it breaks it or how it breaks the protein. And that might tell you something about how the protein works. This is another virus. This is adeno-associated virus. And you can see it also has spike proteins on its surface. And sometimes we use proteins as tools to study other to study cells or other things. For example, green fluorescent protein is a protein that glows green, and scientists use it to study, to be able to see all sorts of things inside the cell. So let's say you want to design a new protein. What do you start with? You start with 20 building blocks called amino acids. These amino acids have this group with the nitrogen, the N and the two H's on the left side. And then on the other side, there's this carbon with 2 oxygens and a hydrogen on the right side. And then there's that R group, which is the part that changes between different amino acids. In nature, there are primarily 20 different amino acids, and they differ in what's in that R group. And scientists often just abbreviate them with a letter. So we have these 20 building blocks. And out of those, when we build a protein, we build it one at a time. We get a sequence. Say we start with the M, then go to G, T, G. And once you have a whole sequence, that sequence folds into something, a structure, a 3-dimensional structure. And that structure then determines what that protein can do. So in this case, you have some sequence and it folds into that spike glycoprotein, which allows the coronavirus to infect lung cells. So the protein design problem then comes down to, given the thing you want the protein to do, say I want a protein that will break down PET, which is plastic, used in bottles, how do I get the sequence of amino acids that will let me do that? And even though scientists have been working on this problem for a long time, you can't just write down what you want the protein to do and print out the sequence of amino acids that would do it. And if we think about how hard this problem is, there are 20 amino acids. If I have a protein that's length 200, there are 1.6 times 10 to the 260 possible length 200 proteins. That's a really big number. For an idea of how big, there are only 10 to the 82nd atoms in the universe. So there's not even close enough atoms in the universe to try all the proteins at length 200. To make this even better, most proteins that people try to design or engineer are at least 200, sometimes 500 amino acids. If you've ever heard of CRISPR, which is a very famous protein used now for editing DNA, it's several thousand amino acids long. So the space of possible proteins is just humongous. It turns out that if you find a way to get around this problem, they give you a Nobel Prize. So my PhD advisor, Francis Arnold, is famous for coming up with something called directed evolution. And the idea here is we don't know how to design proteins, but nature has had a couple billion years to design proteins, and it seems to have done a fairly decent job. So why not just use nature's methods for designing proteins? But what you do is you find some protein that does the thing you want to do, but not very well. Then you take it and you change it or mutate it a bunch of times. And now you have a whole bunch of variant proteins that are just a little bit different from the parent. Then you measure how well or screen how well each of those variants does the function you want. You throw away all the ones that aren't better than the starting point or the parent. And then you use the one that's the best as the new parent, and you repeat this until you're happy. But sometimes that screen is really hard to do. You can imagine, what if I want a protein that is something that's very hard to measure? Then I may not want to screen thousands of variants. Or sometimes we don't even have a starting point. How do we get around that? So most of my work really focuses on the cases where you can't just use a simple method that doesn't require much knowledge. And I use a tool called machine learning. And machine learning is a bunch of mathematical and computer science methods for learning patterns from data. What I mean by that is, traditionally, when people program a computer, you take your knowledge and you put it into the computer such that, given some inputs, you can use your knowledge, or the computer can use your knowledge to spit out some predictions or some outputs. What machine learning does instead is you start with inputs and outputs. So inputs here can be protein sequences, and outputs can be how well that protein does the thing you want to do, their function, or why. And then you what's called train your model, so that your model learns the patterns from your observations. Then once you've trained your model, you take your model and give it new sequences, and it predicts how well those sequences will do the thing you want to do. So what this kind of looks like is, you can think of a plot of sequence versus function, and you observe those black X's are things you've observed, sequences, how well they do the function you want, and then you use those to try to predict how every other sequence will do the thing you want to do. So one way we could use this information is going back to this directed evolution. Instead of throwing out all the data from the sequences, from the variants that don't improve, use all of that, train a model, and then use that model to choose new sequences and repeat. So that's kind of like, if this is all the possible proteins in the world, nature has seen some very small number of those possible proteins. And we're going to start from those and use machine learning and just push the boundaries of what nature has seen. But I want to be a little bit more ambitious than that. I want to go out and skip away from what nature has seen, out into the space of all possible proteins, and find things that nature has never seen. It's nothing like what nature's seen. So to do that, the idea here is we want to generate proteins or hallucinate proteins. So I can go and people have written down and stored somewhere, not all, but many natural proteins and what they do. I'm going to use that to train a machine learning model, and then hopefully I can take some new function that isn't seen in nature, and my model will spit out or generate a protein or many proteins that would do that are far away from the natural proteins. So that's kind of the goal of my research. And that is the last slide, so I'll take any questions now.
Thank you very much, Kevin. I have a question for you. Right now, for protein engineering, what would be the highest impact applications that in your mind?
Yeah, the highest impact applications, I think, are in environmentally friendly processes. So a lot of the important chemical processes that our society relies on, for example, making plastics or making all sorts of other chemicals, use very unenvironmentally friendly methods. So if we can do those with proteins which are biological, they're renewable, they don't use environmentally damaging chemicals, that would be very high impact. The other general high impact field is in pharmaceuticals, so designing new medicines. For example, I think every pharmaceutical company on the planet right now is trying to design a drug for the coronavirus for good reason. And many of those drugs are either proteins or they target that spike protein or some other protein on the virus.
Yeah, very interesting. Thank you. And we have another question about how do the proteins, or how do you mutate the sequences to generate all the proteins?
Yeah, so that's actually like a whole hour-long talk we could have. So some things you could do. But the simplest way is just, so in a molecular biology lab, the way scientists have designed ways to start from DNA and then reproduce that DNA many, many times. And then if you have the DNA, you can put it in the cells and the cells will turn into protein for you. The simplest way is just to take that process of reproducing that DNA many, many times and make it a little less good so that it makes some mistakes. And then those mistakes will change the DNA, which changes downstream protein. That's the simplest way. Other ways are you can choose where in the protein you want to mutate. And you can just order the DNA that will give you all the possible amino acids at that position. And you can do that for a few positions if you want. And then there's more complicated ways that involve like shuffling things or chopping your proteins up and chopping different proteins up and shuffling things around. But those are probably the three major ways to do random mutagenesis.
Great. And I think we have time for one more question. This is a more technical question about your machine learning.
Mm-hmm.
How do you know that you found a global maximum and not like a local maximum in your analysis?
I mean, you never know unless you can test every point, right? And if you can test every point, then you just test every point and say you found the global maximum and forget the machine learning.
And can you shortly or briefly explain what the maximum means in your analysis?
Right. So if we think of say, I want a protein that will glow really brightly, right? A local maximum would mean that given my starting point, so local in this case we'll say means proteins that I can get by editing one amino acid at a time from where I started. And so in that little region, I might come to a protein where no matter how I change it, it always makes it worse. So that's a local maximum. The global maximum is just the brightest protein possible. And given the more than the number of atoms in the universe, number of proteins, it's highly unlikely that we'll ever find the brightest one. So the question then is, have you found one that's bright enough for your purposes?
Yeah, that's a great answer to a highly technical question. All right, that's all the time we have for questions now. If you have further questions, we may be able to answer them offline. Thank you. And thank you, Kevin.
You're welcome. Thank you.
Our next speaker today is going to be Arun Nagpal, who is a graduate student in the Applied Physics department. In particular, he's working in the group of Professor Harry Atwater. His work at Caltech focuses on the study of surface plasmons, is one of the most promising materials of the future, that is graphene. Arun will make a great job explaining all these concepts to us, so thanks for being here today, and I'm handing it over to you.
Thanks so much. Let me get started here. There we go. All right, so thank you so much for that introduction. My name is Arun, and I'm a first-year graduate student. And as mentioned, I'm going to talk about plasmonics. So let's get started. So as an applied physicist, so much of my field is dominated by the questions concerning light-matter interactions. So in a nutshell, broadly speaking, light-matter interactions can be described as the series of phenomena that occur whenever you have light, or more generally, any kind of electromagnetic wave interacting with matter, stuff. Whenever they come into contact, what happens? So schematically here, you have on the left side a representation of an electromagnetic wave. And in this case, all the light that I'm going to be talking about is going to be like laser light. So it's going to have a well-defined wavelength. And for those of you that don't know, wavelength is a parameter that tells you what color the light is. So for example, red light has longer wavelength than green light, which has longer wavelength than blue light. On the right side, you have a couple of different forms of matter, right? So we're surrounded by matter. Matter is entirely made up of atoms, but those atoms can be arranged in multitudes of ways. So the top two photos on the right side consist of a crystal structure where the atoms are sort of ordered in this regular array. And this material is called hexagonal boron nitride. You can see it's these alternating nitrogen and boron atoms arranged in this hexagonal structure, hence the name. On the right side, you see, actually, this is actually a photograph of hexagonal boron nitride. It's a microscope image taken of the atomic structure where you can actually discern, if you kind of squint, the hexagonal structure of the atoms. This is a photo taken by a colleague of mine who works in the same lab as me, and I think it's just incredible how we have the tools and the technology to take photos of atoms now, you know? And then this bottom photo is a protein. So I don't know anything about proteins. I think we've all learned quite a lot from our last talk. But I want to point out that the main difference that's relevant here is that proteins have this very complex structure. So of course they're still made of atoms, as we discussed a few moments ago, but you know, proteins have all this tertiary structure. They have spirals and tangles and are very complex in that regard. And what makes light such a valuable tool for understanding matter is that light interacts with the different structure of different materials in a variety of ways. And by studying the changes as material structure changes, we can understand a lot about the structure and characteristics of materials. So a lot of the light that, a lot of the materials that we're going to be talking about today are actually metals, specifically noble metals. So some examples of noble metals, as you can see, are gold and silver. Some other examples you might be familiar with are platinum, palladium, iridium, a lot of ioms. And what makes them noble metals specifically is that they resist rusting or oxidation. So gold is one of the most inert metals that we know of. And you might notice your gold jewelry tends to stay shiny for a long period of time. And that's partially due to gold being resistant to this kind of oxidation. Silver, on the other hand, is still quite oxidation resistant, but not as much as gold, which is why silver tends to tarnish faster. Right? So here's your first major vocabulary word of this presentation, free electrons. So all metals are composed of these charge-carrying particles that make up electricity, electrons. And metals in particular, their electrons, they have a huge population of electrons that are sort of not really strongly bound to the constituent atoms of the metal. So they are free to move around, subject to external forces. One such external force might be light. And so if you apply an electromagnetic field, like light, to these materials, the free electrons inside of these materials are going to move, right? And so this is the key sort of physical insight that we need to understand what plasmonics is. So what actually happens when you shine light on metals? Well, let's consider a model where we have some light with a defined wavelength. We're shining a laser onto a chunk of metal that's really, really small. That's only a few tens of nanometers wide. We call this a nanoparticle because it's nanoscale. And for reference, a human hair has a width on the order of 10,000 to 150,000 nanometers. So something that's a few tens of nanometers wide is really, really, really small. So a lot of the physics that we're interested in occurs when the wavelength of the light that we're exciting our nanoparticle with, that we're shining onto our nanoparticle, is much, much larger than the width of the nanoparticle. So a more accurate diagram might look more like this, where the light that we're exciting has this enormous wavelength relative to our nanoparticle. So what actually happens when we have this light, let's say it's propagating left to right through this nanoparticle? What happens to the electrons inside that nanoparticle? Well, the free electrons are going to move. They're going to move in response to the electromagnetic field of the light. And since the field of the light oscillates in time, the electrons are also going to oscillate in time. And so this blue thing that you see moving back and forth is like the electron cloud. It consists of all the entire population of free electrons inside this nanoparticle moving in unison, sort of sympathetically with this oscillating light field. And this is what we call a localized surface plasmon resonance. So if we tune the wavelength of the light just right, we can see that we get what's called a resonant response, where the scattered light from the material actually is of a different energy, is a different color than the light that we excited with. So this is what composes the field of plasmonics, the study of these types of excitations. So let's be more concrete about what that actually looks like. This is a 1600-year-old Roman goblet that is made out of this special kind of glass that has embedded in it a lot of these very small gold nanoparticles. And it turns out these gold nanoparticles are about the right size to observe this kind of localized surface plasmon resonance. So the left image you see here is a picture of the cup when it's illuminated from the outside. When it's just sitting out and you've got fluorescent light above you and you're just looking at it, you see this kind of greenish natural color of the glass. The right image is the cup illuminated from the inside. And what this changes is the fact that the light now has to propagate through the extent of the walls of the cup. So it has to pass through the cup to get to your eyes. And in doing so, it interacts with all of these little gold nanoparticles, producing this localized surface plasmon resonance and scattering off this red light, this different colored light. And that's why this cup will appear like it's a different color depending on where your illumination is coming from. And that's a really cool and unintuitive thing, right? So that's one of the notion of plasmonics has been around for a long time. We've been using this technology for millennia. Stained glass windows work in a really similar way. So, but let's talk about concrete applications in modern technology. So my group published this paper a little over a decade ago concerning improving the efficiency of solar cells by incorporating nanoparticles into their construction. So solar cells turn light into electricity. This image on the left is just a schematic of the solar cell design, where we're exciting from the top, we're illuminating it from the top, and we've decorated the top with a bunch of nanoparticles of different types. And we want to study how the solar cell efficiency improves based on the type of nanoparticles that we add to the solar cell. So on the right side, we have a plot that is, it looks pretty complicated, but we're going to get through it together. So the key insight here is that the places I've marked with red arrows are where the, if the curve moves in that direction, the solar cell becomes better, right? So we want the curve to move roughly in the direction of the red arrows. So this black line that you see on the right, I'll use my pointer actually, this black line that you see on the right corresponds to the solar cell with no nanoparticles added. This is our control. And then all of these colored lines correspond to the solar cell when you add different kinds of nanoparticles to it. And you can see that one is slightly better than all of the others, this blue line up top. And so this is like a comparative study showing us how this localized surface plasmon resonance actually allows us to trap light with more efficiency than we would if the nanoparticles weren't there. And this is really important. This improves the efficiency of solar cells. It means that you can get more bang for your buck when you buy a solar cell, and that'll improve, adoption of renewable energies and improve the feasibility of solar cells being used all over the world. So my current research project concerns plasmons in graphene. So graphene, for those of you that might be subscribers to the Scientific American or pay attention to science news, graphene consists of this hexagonal array of carbon atoms. You might be familiar with. So like hexagonal boron nitride, graphene is this hexagonal array, and it's known, or rather famous, for being one layer thick, one atomic layer thick, and having very interesting chemical, mechanical, and optical properties just in that one atomic layer. So my experiments have been involved exciting graphene with some laser light, generating a plasmon. And this time the plasmon isn't actually localized. It isn't confined to a nanoparticle. It's in this big plane. So it actually propagates. So this arrow represents a plasmon excited by this light propagating through the graphene. And eventually it is destroyed. It relaxes and gives off light. And the interesting thing about the light that it gives off is that this light is actually in the mid-infrared. So it's the light that we can't see, but normally feel as heat. One really interesting potential application of this work is that we can use this mid-infrared light to make a laser device out of this graphene. And mid-infrared lasers are actually very, very difficult to make at present. They're usually very complicated. You know, the materials to make mid-infrared lasers are very hard to manufacture. And so to be able to do it in graphene would be enormous. You could use this laser for materials characterization, for narcotics detection, for remote sensing. Infrared light has a lot of really interesting properties in medicine. It passes through the skin easily, so it could lend itself to non-invasive medical procedures. The list of applications for a mid-infrared laser is, especially a small one, are endless. So I want to close out my presentation by showing you some data. Because I'm a first-year graduate student. I collected some data. I got really excited about it. Well, I simulated some data and I got really excited about it. And I want to show you guys. So on the left here, you see a cartoon of sort of how I want to set up the experiment. This gray sort of plane is the single layer of graphene that I'm trying to work with. This arrow marked by KSP is the plasmon moving through the graphene. And then you see these little yellow disks. And these are actually gold nanodisks. And like I discussed with gold nanoparticles, when you excite these nanodisks with a laser, you can generate a localized surface plasmon resonance. And so when the plasmons in the disks meet the plasmons in the graphene, what happens is you get this emission light that's really, really bright. That's much brighter than if these nanodisks weren't here. And so on the right here is a simulation that I did recently showing the response of these nanodisks to a laser. And I wanted to see a localized plasmon resonance. And in fact, it does look kind of like the little cartoon I showed you earlier. And so I think that's really promising. As soon as I can get back into lab, this research will be ongoing. That's the gist of plasmons and plasmonics. So that's my presentation. I'd like to acknowledge John and Pablo for having me here, and I'd also like to shout out to my research group for supporting me and being all around just fabulous. So this picture on the right isn't my research group; it's my research group plus alumni plus collaborators and friends, but they're all in there somewhere. So thank you so much for listening. Happy to take questions.
All right, thank you, Arun. We had a question about the cup that you showed. Mm-hmm. So you had it illuminated from the inside. Mm-hmm. So if you were to if you could change your view to the inside of the cup and had the light source outside, would you see the same colors? Yeah, you see it as red, because the relevant factor here is that the light has to pass through the cup to generate enough scattered red light
for you to see. Yeah, so if you were inside the cup and the source were outside the cup, I believe, yeah, you'd be able to see basically the inverse of what you see here. The pattern would be mirrored, but you'd see it as red.
And we have another question about the graphene. So does the size or the depth of the graphene make a difference in the generation of plasmons, plasmons? But is it just a surface effect, or does it matter the bulk underneath that surface?
Oh, that's actually a really, really good question. It's a really subtle question. So the bulk does matter insofar as we're dealing with a monolayer system. So it's one atomic layer is thick, but it's sitting on something. And depending on what it's sitting on, actually the number of free electrons in the graphene actually changes natively. And we can adjust that using external forces, but naturally the physics of it changes when you change your underlying substrate, your underlying platform on which your graphene is mounted. So that does change the energy and the lifetime, so the amount of time that the plasmons spend propagating of your plasmons. And that will really affect how efficiently you're able to get light out of the system. Absolutely.
Great. And we have one more question. What makes graphene a better material than other potential materials for this application?
Yeah, that's again a really good question. So before I started this work, as is common in a lot of sort of the research pipeline of physics, a lot of theorists were interested in the prospect of using graphene as a laser. And a really important characteristic for creating lasers out of materials, whether it be graphene or anything else, is that you generate what's called a condition called population inversion. Now, it would be really difficult to really get into the details of how lasers work right now, but what I can say is that you have to be able to excite all of your free electrons to a really high energy. And once you do that, you create a condition called population inversion, and that produces the laser effect. That's what makes lasers different from, say, incandescent light bulbs or fluorescent tubes. And so what makes graphene interesting is that you can actually apply a battery to graphene and change how many carriers you have at your disposal to work with. And by changing that number, you actually change how easy or difficult it is to generate this population inversion condition. What this means is that you have this degree of tunability that's really, really exciting. Also, graphene plasmons are in the mid-infrared, and so mid-infrared being a regime of light that just hasn't been really properly taken advantage of to generate laser devices. Being able to generate plasmons that strongly emit in the infrared is really valuable, and that's one of the things that makes graphene interesting for this as well.
Great. Thank you, Arun.
Thanks so much.
Okay, our last speaker today will be Celeste Labedz, who is a graduate student in the Department of Geological and Planetary Science. She's working in the Seismo Lab, where she's using seismological tools to study environmental processes related to glaciers. Apart from that, Celeste also likes Disney movies, as you might have imagined from the title of the talk. Celeste, thanks for being here today. We are excited to hear from you, especially those who, like me, are also fans of Frozen.
All right, let me get my presentation up here on full screen. Okay, can we see that?
Yes, perfect.
Stellar, let's go. All right, so thanks for tuning in, everyone. I know that a lot of us, me included, are streaming plenty more movies than usual while we're all staying home during the COVID pandemic. So I thought it would be fun to bring some of that movie magic to our talks today at Explore Caltech. So I'm going to be talking about some real science of glaciers in the fun lens of Disney's Frozen 2. So there's way more science to talk about in the Frozen movies than can fit in any 10-minute talk. Even Disney had a team of scientists work with their animators to get the snow physics right in the first movie. So today, I'm just going to be touching on a few topics about glaciers here that we can see during one scene of Frozen 2. And don't worry if you haven't seen Frozen or Frozen 2 yet, you can still get all of the science and I'll catch you up to set the scene in the movie, and there won't be any big spoilers in case you want to watch it later. So first off, I'm going to introduce myself. I'm a PhD student in geophysics at Caltech Seismolab. My research is all about glaciers. I've spent 2 summers doing fieldwork on glaciers in Southeast Alaska. So for my research, I put seismometers, the same instruments that detect earthquakes, onto glaciers to observe the motions that happen inside of the ice. And I also really like Disney movies and all kinds of fun kids media. And I think that Queen Elsa from Frozen, with her talent and passion for ice, would actually be a really awesome glaciologist. And I wanted to know what glaciologist Elsa might look like, so I decided to just try being her myself. I brought a cape and braided my hair at my fieldwork, and I got this photo, just so we can imagine what kind of science princess would be investigating this type of thing. So first off, let's get to the movie magic. I gotta set the scene for you so we all know where we are. We're gonna be following the song Show Yourself from Frozen 2. Now, if you're watching the video afterward instead of the live stream, this would be a great time to pause and check out the sing-along video on YouTube, because the full scene from the movie is there. And I'm gonna be using scenes from that music video, snapshots there, to guide us as we go. So here's the catch-up if you haven't seen Frozen 2. Queen Elsa is looking for the secrets of her past in order to save her kingdom from danger. And she decides to go looking in Ahtohallan, which is a place described as a river full of memory in a lullaby that her mother used to sing to her. And Ahtohallan is a great place for us to learn about glaciers. So I think we should go there and check out what we can learn. So just like Elsa, I hope you are ready to learn too. So let's take a look at Ahtohallan as we approach. This is when Elsa is riding her water horse towards this mystical place, and we get our first view of Ahtohallan. And when I was watching this in the movie theater, I thought, that's a glacier, I'm so excited. So now, if you haven't seen a glacier before or don't know what they look like, here's a side-by-side view of Ahtohallan in Frozen and Fjalsjökull, a real glacier in southern Iceland. And you can definitely see the similarities. They look the same. Disney did a good job of making a pretty realistic, although a little bit dramatic, glacier for their movie. So we know that they called Ahtohallan a river full of memory, but this doesn't quite look like a river at first glance. This is a humongous pile of solid blue-white ice just draping down a mountain. It doesn't seem very rivery. So let's explore that river idea by learning how glaciers are made first. Then we'll see why it's a river. So here's a cross-section diagram of a glacier. Like we just sliced right down the middle and are looking at one side of the slice. So the only thing that you need to make a glacier is snow. You need a lot of snow, but really all you need is snow. What you need is more snow to fall every winter than will melt every summer. And this doesn't happen everywhere. This only happens in very cold places that get a lot of snow. So that's like at the very tops of high mountains and near the North and South Pole. Those are the kinds of places where you can get glaciers. So with that excess snow, it piles up. Year after year, we get more and more snow piling up. And it starts to get heavy after you have a lot of it. Even though a handful of snow seems light, it can really add up when you have, hundreds of feet. So the snow at the bottom starts to get squished by the snow at the top. And eventually it gets compressed from nice fluffy snow into very solid ice. And that weight does even more than just compressing it into ice. It can actually make the ice move. It presses so hard when you have hundreds and hundreds of feet of snow, like in real glaciers, that it can make the ice flow like it's a liquid, even though it's a solid, as we know. So it can be kind of weird to think about a solid flowing, but you can kind of get an intuition for it by thinking about other solids that flow a little bit more easily. Like if you have softer modeling clay or like slime, those, if you make a ball out of them and just set it down, it'll sort of ooze back into a blob just under its own weight. It didn't need extra force on it. So when you have enough ice, it flows just the same. So a glacier will just keep flowing, and it'll eventually reach somewhere, maybe lower elevation down the mountain, or maybe lower latitude and a little bit warmer, where it won't be accumulating snow anymore. And when it gets down there, then the ice can start to melt or break apart, and that's where the glacier will reach an end. So this is how you make a glacier. And now that we know that, we can see that glaciers really do flow like rivers. It's from that weight of the snow on top, and all of that ice causes it to flow downhill. So as Elsa approaches Ahtohallan, she makes this realization too, and says, of course, glaciers are rivers of ice. Now, if we look from above, we can really get this river comparison in our heads. So here's two images from space. These are satellite images. And one of them shows some rivers, and the other one shows some glaciers. They're both generally flowing from right to left on here. And you can see a lot of similarities. They're flowing downhill, down valleys. They squiggle a little bit as they go, and there are branches that join together. So in this view, you can really see the river part of a river full of memory. So the lullaby that Elsa's mom sang was already, we know, half right. Now let's check out the other half of that line of the song, a river full of memory. So real glaciers actually are full of memory too. This is a really awesome bit of science, because scientists use the memories that glaciers have to help understand our planet's past. So remember how glaciers are formed from the extra snow each year? That means that the ice is in layers, and each layer is 1 year, kind of like the rings in a tree. So in a glacier, the oldest ice is at the bottom, and the youngest ice is at the top, where the freshest snow is. Now, scientists want to get a nice cross-section of these different ages of ice, so they drill for cores. This is a picture of an ice core. So that's just, you drill down and you pull up a cylinder of ice that has a little bit of each layer in it. And so having all those layers is kind of like having a scrapbook of the glacier's history. Each layer contains information about what the world was like when that snow fell. There can be bits of volcanic ash or plant pollen that blew in on the wind. Or there can be bubbles of air that got trapped when the snow fell and then was compressed into ice. Even the chemistry of the ice itself can give clues about the past, especially about the climate. Now, ice cores are some of scientists' best tools for understanding the recent past of our planet, especially for things like climate. Some of the oldest ice ever found is in Antarctica, and that's over 2 million years old. And glaciers all around the world have their own records of the past. So it's a really cool way to see what the world was like over the past several 100,000 or even millions of years. So there we have it. The song is right on both counts. A glacier really is a river full of memory, just like the song in the movie says. Now, of course, the memories that Elsa was looking for in the movie in the glacier are a little bit different than what scientists are looking for in the glacier, but it's still the same cool idea of finding the past hidden in the ice. Now, the next cool bit of science that you can see in the show yourself scene of the movie is the tunnels inside of Ahtohallan Glacier. It may seem like movie magic as Elsa dashes and slides through these like glittery blue tunnels, but this part is actually surprisingly real, and it's really awesome. So liquid water from rain and melted ice moves through the inside of glaciers in caves and tunnels, a lot like we see in Atahalin. So you can get water on the surface of glaciers when it rains on them or when it's a warm sunny day and some of the surface melts away. And that water on the surface will flow downhill, just like any water would. And it can sometimes fall into these dark, scary holes called moulins, where the water can enter the glacier's sort of internal plumbing. And then once it's inside of the glacier, the water will move around in cracks and tunnels and caves. It really is, it's like plumbing inside of the glacier. And that can be either inside of the ice or between the ice and the rock that is below it. So that water keeps flowing downhill and eventually comes out near the end of the glacier. When there's less water flowing through the system, it becomes less like water pipes and more like empty caves and tunnels of ice, just like in this picture, and just like Elsa goes into in Atahalin. So now, just like Elsa, we have come so far in learning some fun facts about glaciers. And Elsa was looking for that knowledge for a good reason when she went into Atahalin. She was looking for a way to save her kingdom. And we can use knowledge about glaciers to help save our world too. So in our warming climate, the world's glaciers are shrinking because more ice melts every year than can be replaced by new snow. And losing those glaciers can cause sea level rise, which is really hazardous to coastal communities. And communities who are downstream of high mountains, they rely on glaciers steadily melting throughout the summer to get fresh water for their agriculture and drinking and washing and all their water needs. So it's really important to have glaciers stay existing in our world. And we can't cast a spell to cool down the world like Elsa can, but the next best thing in ice magic is to educate ourselves on climate science and to elect leaders who do the same. So if you would like to show yourself a little bit more about the science of glaciers, you can take a screenshot here if you want, or pause the video and check out these links. So yeah, thank you so much for listening. Next time you watch Frozen or Frozen 2, you will know a lot more about the real science of glaciers to go with the movie magic of Atahalin. And yeah, now I'm happy to take any questions that folks have.
Thank you, Celeste. I think we have time for maybe one or two questions. The first is, you said the ice preserves, or you can learn different things from the ice, including like gases that are trapped. What about biological items like pollen or bacteria? Do you learn, is there anything like that trapped in the ice, and is that well preserved?
Yeah, so biological material like pollen can blow in and sometimes be preserved in glaciers. You can usually find these more likely in mountain glaciers where they're nearer to vegetation than somewhere like Antarctica. But yeah, those can give you a clue of what kind of plant life was around at the time when that layer of snow was laid down. And even bigger biological material can be preserved in glaciers too. Sometimes larger living things have been trapped in ice and then found later after they flowed through the glacier and melted out the other end.
Great. I think that's all the time we have for questions. Thank you very much, Celeste.
Thank you.
Well, today we had a great session with fantastic talks. And first, we want to thank again all the three speakers for sharing their time and bringing us a little bit closer their amazing research. And secondly, we also want to thank all the audience that joined us today. We are excited to see this level of participation, and we hope you all enjoy this event as much as we did. Before leaving, I just want to remind a couple of things. So first is that this online version of the Explore Caltech, as John mentioned in the introduction, is going to be divided into different sessions. So after this first one, we invite you all to join us again next Wednesday, May 13, same time, send some link for our next session. We will send anyway further emails with that information so that you can easily join. And secondly, we want to mention that the recorded video of each session will be posted in the website of Explore Caltech at explore.caltech.edu, where you can also find information about the next talks. So as I said, I hope you all enjoy this event, and I hope you have also a nice week and stay safe. Thank you for coming.